morning. How are you doing? Thank you so much. Now we'll continue our discussion of nucleic acid metabolism. But before we start, it would be best if we review our previous discussion on this nucleic acid metabolism. Let's see. I will review a bit about this nucleic acid metabolism. We have discussed already nucleotide as DNA and RNA building block, dietary nucleic acid metabolism, and Purin nucleotide biosynthesis, and pyrimidine nucleotide biosynthesis. In this case, we discuss about the anabolic reaction. Now we will continue to Purin degradation, pyrimidine degradation, and metabolic disorder. In these two subtopic, it's related to catabolic reaction. Now let's move to catabolism, Purin and pyrimidine. See this one, Purin catabolism. All Purin degradation lead to uric in a pyramid. Normal rate of uric acid excretion by human is about 0.6 gram per 24 hours in urine. Normal concentration in the serum is 3 to 7 milligram per deciliter. And nucleotides, this is a group specific, and phosphatase non-specific, degrade nucleotide into nucleoside. Let's see this reaction in the down side of this slide. This one nucleoside hydrolyze and convert into base and ribose. And this reaction catalyzed by nucleosidase. And the second one is a nucleoside phosphorylase and will produce base plus ribose 1 phosphate. In this reaction, this will be catalyzed by phosphatase. Then let's see the intracellular Purin metabolism. Xanthine is the point of convergence for Purin-based degradation, and this xanthine will be broken into uric acid. Purin and pyrimidine ribonucleotide degradation pathway are the same for deoxyribonucleotide, and we should remember that two key enzymes involved in this intracellular Purin metabolism. The first one is nucleosidase and Purin nucleoside phosphorylase, except for adenosine. Ribose 1 phosphate can be converted into PRPP for de novo EMP synthesis. Let's see the structure of xanthine in the right side of this slide. And this is xanthine. Let us see this one, the structure. Then let's move to adenosine degradation. IMP will be deaminated into EMP by IMP deaminase. IMP and EMP are both converted into adenosine and inosine by nucleosidase. Adenosine is deaminated into inosine, and inosine is further converted into xanthine. Let's see this pathway. This is AMP. This AMP will be converted into EMP by catalyzing with this enzyme AMP deaminase. Then this EMP will be converted into inosine, catalyzed by nucleotides. On the other hand, this EMP will be compared into adenosine catalyzed by nucleotidase, and this adenosine also will be compared to inosine. Then this inosine will be compared into hypoxanthine. Then this hypoxanthine will be compared to xanthine. On the other hand, these hypoxanthines can be compared into Purin sulfate pathway. So let's review once again about this adenosine degradation pathway. AMP will be deaminate into EMP by AMP deaminase. AMP and EMP are both compared into adenosine and inosine by nucleotidase. Adenosine is deaminate into inosine, and this inosine is further converted into xanthine. Then let's move on to this adenosine degradation XMP pathway. The first one is deamination of AMP. In this case, the enzyme involved in this reaction is AMP deaminase. And the second one is NAD-dependent oxidation of EMP, and the enzyme involved in this reaction is EMP dehydrogenase. And the third one is dephosphorylation. Enzyme involved in this reaction is nucleotidase. And the last one is for phosphorylases. The enzyme involved in this reaction is xanthine phosphorylase. Let's see this one. We start from this molecule. This molecule will be accompanied into EMP. Then let's see this step 2. Step 3 will accompany to xanthosine, and xanthosine will be compared to xanthine. Then let's move to guanine degradation. Xanthine is the deaminate form of guanine. Let's see this one. This is a guanine molecule, and this one is a xanthine molecule. Guanine is compared into xanthine by guanine deaminase or guanase in the presence of water. This one. A guanine is deaminated in its free form, unlike adenine. In this case, this one are still attached to sugar. Then now let's move to xanthine degradation to uric acid. Comparison of this hypoxanthine to xanthine and xanthine to uric acid are catalyzed by xanthine oxidase. And this one are xanthine oxidase. This is a xanthine. Xanthine oxidase is a homodimeric protein that generate reactive oxygen species. And the last one, hydroperoxide, will be disproportionate by a catalyze. By catalase. This is an enzyme. See this one again. This xanthine will be compared into this uric acid. Let's see this structure of 3 molecules in this downside. This one is a hypoxanthine, and this one is a xanthine, and this one uric acid. In hypoxanthine molecule, it has 1 oxygen in it, and in xanthine, it has 2 oxygen, and uric acid, it has 3 oxygen in this case. Then now let's move to species-dependent excretion of nucleotide. Human and other primate X-ray nucleotide in form of uric acid, along with other species such as bird, reptile, and insect. Other organisms are capable to undergo a further degradation of uric acid. Now let's see this pathway. In the right side of this slide, this is uric acid. This will be excreted by primate, a bird, a reptile, and insect. Let's see this one. The first one, uric acid, will be compared to allantoin. In this allantoin, this ring will be open. This one, see this one. To compare into this molecule, then this allantoin will be compared into allantoic acid. In this reaction, we can see that this ring also will be clipped and open into allantoic acid. From this allantoic acid will be compared to urea by catalyzing with allantoicase, and this urea will be compared into ammonia. Now let's see what happened with this urea and ammonia. This pathway will be occur in cartilaginous fish amphibian, and this one will be occur in marine invertebrate. Now let's see Purin degradation overview. See this one, AMP, EMP, XMP. AMP will be compared into adenosine, then this adenosine will be compared to adenine. In this EMP will be compared to inosine, then this inosine will be compared to hypoxanthine. SMP will be compared to xanthosine, and then will be compared to xanthine. Let's see this one. From this one, those three molecules will be compared into xanthine. Then let's see this one, GMP also will be compared into guanosine and guanine, then will be compared into xanthine. Then the xanthine will be compared into uric acid. Now let's see the line in this left side. This is deamination. See this one. Step 1 is deamination, and this one oxidation, and this one phosphorylation, and this one is a phosphorylasis. Now let's move on to pyrimidine catabolism. CMP and OMP degrade to base similarly to purin by deamination and glycosidic bond cleavage. Cytosine and uracil degradation with beta-alanin, ammonium ion, and carbon dioxide. Beta-alanin can be compared into malonyl coenzyme A to a fatty acid for energy synthesis, or recycled for coenzyme A synthesis. Thymine degradation with beta-amino isobutyric acid, ammonium ion, and carbon dioxide. Then let's see now cytidine and uridine degradation. In discussing the cytidine and uridine degradation, we can see that cytidine can be broken down to uridine by cytidine deaminase. This one, uracil will be produced by uridine phosphorylase and will be compared into dehydrouracil by dehydropyrimidine dehydrogenase to produce a β-alanin. Now let's repeat once again. This is a CMP, this is OMP. This CMP will be compared to this cytidine, and this OMP will be compared to uridine. And from this uridine will be compared to uracil. This uracil will be compared to this molecule. Then this molecule will be hydrolyzed to open this cyclic compound to produce ureodopropionate, and this ureodopropionate will be compared to a β-alanin. Now have a look to thymine degradation. Thymine is the methylate form of uracil and degraded with the same pathway as uracil. The degradation product of thymine is beta-amino isobutyrate, ammonium, and carbon dioxide. Let's see this one in the right side. This is uracil, and this one is thymine. Uracil will be compared into this dihydrouracil, then this dihydrouracil will be compared to beta-alanin. In discussing thymine will be compared into deoxythymine, and this deoxythymine will be compared into beta-amino isobutyric acid. Then now let's move to conversion of beta-alanin to fatty acid and coenzyme A. Let's see this one, this reaction. This beta-alanin, and this one is malanoic semialdehyde. Beta-alanin will be compared into malonate semialdehyde, and this malonate semialdehyde will be compared into acetyl coenzyme A. In discussing this malonyl semialdehyde, this one will be compared to malonyl-CoA, and this malonyl-CoA will be used for fatty acid synthesis. So now let's conclude once again that beta-alanin can be compared into malonate semialdehyde that will be further broken into acetyl-CoA and malonyl-CoA. Acetyl-CoA will be utilized in metabolism pathway, and malonyl-CoA will be compared into fatty acid for energy use. Then now let's move to metabolic disorder. In discussing this metabolism disorder, we can figure out the several metabolic disorder. In this case, the first one is gout. Second one is Nihana Syndrome, Purin autism, and ADA deficiency or SCED. Now have a look to this one. We'll start with a gout. In discussing this gout, we can see that impaired excretion of overproduction of uric acid, or we call the hyperuricemia. Hyperuricemia is a condition that the concentration of uric acid is above 8 milligram per deciliter, and this uric acid is crystallized into joint, gout arthritis, kidney, and ureter. According to the cause, then this gout is divided into 3 categories: primary gout, secondary gout, and renal gout. Primary gout accounts for 90% of cases. Now let's see this one, gout attack location. This mark is the most common, and this small one is a common, and this one least common. Let us see the most common is in this part of leg, and a common is in this part And the list are common in this part. Now let's see gout arthritis. We will discuss in this case characteristic of this gout and a risk factor. In discussing this one, let's see characteristics of the gout or is intense joint pain, lingering discomfort, inflammation and redness, and limited range of motion. And this factor of this one is obesity or high body mass index, hypertension, the high in meat and seafood, and excess of alcohol use. Let's see this one. This one. Then let's move to gout causes. You can see in this table, this is a primary gout, secondary gout, and renal gout. Now let's discuss one by one. We'll start from a primary gout. The cause of this primary gout, this is caused by enzyme defect and directly or indirectly increase de novo purin biosynthesis and overproduction of purin nucleotide result in high uric acid level. Now let us repeat it once again. This is caused by enzyme defect, directly or indirectly increase de novo purin biosynthesis and overproduction of purin nucleotide result in high uric acid level. Now let's move to secondary gout. In this secondary gout, this is acquired cause due to pathological state that produce abnormal breakdown of cell releasing nucleic acid. For example, of this one is in leukemia, lymphoma, lung tumor, or radiation injury. And the last, let's see this renal cause. This is, let's see the first one caused by insufficiency of renal system, renal failure, or acidosis, and also reduced excretion of uric acid through urine. And the last one is retention of uric acid in blood lead to hyperuricemia. So let's see, we have three gout category. We call that one is a primary gout, a secondary gout, and renal gout. And the cause of this primary gout and secondary gout and the renal gout completely different. Now let's move to gout treatment. In discussing this gout, so far there are so many kind of treatment to combat this disease. This is a metabolism disorder, of course. So xanthine oxidase inhibitor inhibit production of uric acid and treat a gout. Let's see this one, because in discussing this gout, this is related to uric acid concentration. So once again, xanthine oxidase inhibitor inhibit production of uric acid and treat gout. Allopurinol treatment, in this case, hypoxanthine analog that bind to xanthine oxidase. Let's see this one. This is a purin catabolism, of course. This is hypoxanthine. Then this hypoxanthine will be compared into xanthine, and this xanthine will be compared into uric acid. And this uric acid is urinary excretion, and this is normal endpoint in human. Let's see this one, xanthine oxidase. Then in discussing this one, we should move to the previous one, that we have discussed before. Let's see this one. The xanthine oxidase inhibitor inhibit production of uric acid and go allopurinol treatment hypoxanthine analog that bind to xanthine oxidase. Now let's see the structure of these two molecules that can be used to treat metabolic disorder. This one, hypoxanthine, that's this, see this as structure, and we'll see this allopurinol, this one. So in this allopurinol treatment, this allopurinol will decrease the uric acid concentration in this case. Now let's move to Lesch-Nyhan syndrome or NNS. Sex-linked genetic disorder that affect sulfate pathway of Purin metabolism in male in this case. Defect in production of PRTase causing buildup of PRPP. Then these pre-Purin are not recycled to sulfate pathway, but instead are break down and excreted as uric acid. Separately affect brain growth and development. And then each patient have mental retardation, will self-mutilate, self-harming by beating lips and finger of, let's see like this one, this finger. This is a self-harming by himself, and this one also. You can see this one. This separately affect brain growth and development. Then let's move to this Purin autism. In discussing, in discussing Purin autism, 25% of autistic patients may overproduce Purin, and biochemical finding from this test disappear in adolescent, and urine specimen must be obtained in infancy. In discussing this Purin autism, we can see that pink urine will be appear due to uric acid crystal that may be seen in their purse. And pattern will exhibit classical criteria of autism and will experience several seizure and self-injury episode. Several patients observed to benefit from allopurin day, which made them more alert and sociable. So let's see this figure in the right side, pink diaper syndrome. We can see this one. This is a pink urine due to uric acid crystal. This one. See this one. Then now let's move to adenosine deaminase deficiency. Adenosine deaminase deficiency, or ADA. This kind of deficiency, first one, enzyme that catalyze deamination of adenosine to inosine in Purin catabolism. A defect result in a severe combined immunodeficiency, or we call that one an SCED, one of the first diseases to be treated with gene therapy. ADA deficiency causes accumulation of AMP and DATP, effect growth and multiplication of rapidly dividing cell, also causing problem in immune system, die of lymphocyte. So we can see that SCD kill both B cell and T cell, also natural killer cell. Now let's see severe combine of these problem. This is normal, this is abnormal. In discussing immune system for a normal person, this is a T cell and B cell. Then this condition can fight infection, but in abnormal person, this is immune system. This immune system, this SCD kill both a beta cell and T cell, or also a killer, the natural killer cell. So in this case, this one cannot fight infection. So we can see the difference between the two. I think that's all for this morning. Thank you so much for your attention. If you have any question, don't hesitate to inform me. And again, I would say thank you so much. Take care of yourself. See you then by next class. Thank you.